Have you ever wondered where the genetic information of the cell is stored? Today, we are going to make a journey deep inside the cell, starting from nucleus to DNA. We will start with nucleus and then explore the genetic material inside it, and finally zoom all the way down to molecular level to understand its nucleotides. By the end of this video, you will understand nucleus, nuclear membrane, nuclear envelope, nuclear pore, nucleolus, chromatin, chromosomes, chromatids, centromere, DNA, nucleotides, and DNA, nucleotide bases, A, T, C, G. So let's go inside the cell. Human body is made up of trillions of cells. Now we are inside the cell, and the most prominent structure we can see in it here is nucleus. The nucleus is membrane bound organelle. It is found in eukaryotic cells. It contains most of the cell genetic material in the form of DNA. The nucleus is often described as control center of the cell because it provides information, and it has stored its information in DNA, and it provides instruction for many cellular activities. Now let's take a closer look. The nucleus is not simply an empty compartment. It contains several important structures, like it contains nuclear envelope, nuclear pores, nucleoplasm, nucleolus, and the genetic material chromatin. First, we have nuclear envelope. Nuclear envelope is actually a membrane that is surrounding the nucleus. It separates the nucleus from outer cytoplasm. And most importantly, the nuclear envelope consists of two membranes, inner nuclear membrane and outer nuclear membrane. But the question is, if the nucleus is surrounded by membrane, how do substances move in and out of the cell? The answer is nuclear pores. You can see here, these are the pores. They are the openings that helps to substances to pass from inside the nucleus to the cytoplasm. They regulate the movement of molecules. For example, certain proteins, they can enter the nucleus, while RNA molecules and other materials can be transported out. Inside the nucleus, just like the cytoplasm of the cell, a fluid-like environment is present called nucleoplasm. The nucleoplasm actually surrounds the chromatin and nucleolus, and it provides the environment in which many nuclear processes take place. Look at this dense region inside the nucleus. This is called nucleolus. The nucleolus is actually a region within the nucleus where ribosomal RNA is produced. They are also called as the factories of ribosome synthesis. And ribosomal subunits began to be assembled inside the nucleolus. So although the nucleus is not surrounded by its own membrane, it is an important functional region of nucleus. Now we are going to explore the most important part, that is genetic material, the DNA inside the nucleus. It's not simply floating around as naked DNA. It is associated actually with proteins and organized, and the structure is called chromatin. Chromatin is a complex of DNA and associated with proteins that are found inside the nucleus, and we know, like histone proteins. One of the most important protein in DNA is also a histone protein. DNA wraps around histone protein and helping the very long DNA molecule to become organized and in compact form. And most importantly, this chromatin does not always look the same. When a cell is not dividing, the DNA exists in less condensed form, but before the cell division starts, the genetic material becomes much more and highly condensed, and the highly condensed structures forms a chromosome of X shape. You are looking, this is X shaped chromosomes. Human body has 23 pairs of chromosomes, and each chromosome contains a long DNA molecule, and it carries many genes. Now look at this replicated chromosome. Each identical half is called chromatid. When the DNA has been replicated, the two identical copies are known as sister chromatids, and the sister chromatids are connected with each other by a region called centromere, and the centromere is an important chromosomal region, and it is involved in attachment of structure. For example, these structures helps to move equal number of chromosomes in each cell during cell division. And also, as you can see that at the end of each chromosome there are regions called telomeres. Telomeres actually helps to protect the chromosomes and from being incorrectly recognized as damaged DNA. So simply telomeres actually protects chromosomal ends. Now the question is, what is the importance or what is the important distinction between chromosome and chromatid? Look, before DNA replication, one chromosome contains one DNA molecule. After DNA replication, the chromosome consists of two identical sister chromatids that are joined together at a centromere. So once the sister chromatid separates during the cell division, each one become an individual chromosome. So in simple words, one chromosome means one chromatid. It's before replication, but after replication one chromosome contains two sister chromatids. So after replication, when cell division starts, and after cell divisions and separation during anaphase, the two chromosomes are formed. So far we have traveled from cell to nucleus, from nucleus to chromosome, but what is actually inside the chromosome? Let's zoom in one more time. Inside the chromosome, it is DNA. DNA stands for deoxyribonucleic acid, and DNA is a molecule that stores genetic information in cell. It consists of, you can see here, two long strands arranged in a double helix structure, and but DNA itself is built of small repeating units, and these repeating units are called as nucleotide. A nucleotide, as you can see, is the basic building block of DNA. So the basic building block of DNA is nucleotide, and each nucleotide consists of three components: a phosphate group, a 5-carbon sugar called deoxyribose, and the nitrogenous base. That's why DNA is called as deoxyribonucleic acid. As you can see here, the sugar is deoxyribose. It's a 5-carbon sugar, and it is very important, and it actually made up the backbone of the DNA. And as you are seeing, the phosphate group, it connects the neighboring sugars molecules and help the sugar phosphate backbone to grow. So the backbone of DNA is sugar phosphate, and attached are the nitrogenous bases. Now we have four nitrogenous bases: A, T, C, G. Adenine and guanine are double ring bases, and cytosine and thymine are single ring bases. As adenine is represented by A, thymine is T, cytosine is C, and guanine is G. As you know that DNA consists of two strands, and here you can see these strands run in opposite direction and are held together by their interactions between these nitrogenous bases. The hydrogen bond is found between these nitrogenous bases, and most importantly, you need to keep in mind that always A bind two double bonds with T, and C always form three bonds or three hydrogen bonds with G. The bases do not pair randomly, and it is very specific and always happen in this way: adenine with thymine, cytosine with guanine. This is called complementary base pairing, and together these two complementary strands twist around each other to form a double helix structure. Now let's zoom back out and put everything together. Nucleotides from DNA, DNA with proteins to form chromatin, chromatin highly condensed to form chromosome, chromosomes are located inside the nucleus, and the nucleus is one of the major organelle inside the whole cell, and cells is the basic unit of whole organism. If you want to explore more regarding the topics, Check out the related videos on this channel and don't forget to subscribe the Regal Biology with Khadija for more biology explained through animation.